So we're going to continue with talking about horizontal gene transfer, and I've already gone over the basics of transformation, where there's DNA in the environment and it can be picked up by bacteria and alter its genome. Conjugation, where one bacteria will actually pass DNA directly to another bacteria. Now what we're going to look at is transduction. So transduction is where there's viral DNA transmission. So for bacteria, this is when we have a bacteriophage. This bacteriophage is going to attach to the surface of a bacteria, dock to it. And then what it's going to do is inject its DNA into the bacterial cell. Now, when that DNA is, we're going to talk about viruses and virology and more the life cycles later on, specifically from the viral perspective. Now we're looking at it from the bacterial perspective and how the bacteria gains new genes because of this process. So they're same process, just different emphasis. Now we're emphasizing the bacteria. So for the virus, though, it transmits DNA into the cell. Now what's going to happen is either one of two things. This process will either enter into something we call a lytic cycle or a lysogenic cycle. So lytic or lysogenic. And that will determine what type of transduction we have. So whether we're going to have generalized or specialized transduction. Okay. So we'll get into this. Lytic. What's going to happen here? With the lytic cycle, the DNA that's injected into the bacterial cell is going to start being copied by the cell. In addition to that, genes within the virus are going to be expressed. Some of those genes are going to contain proteins that are going to then cut up the host DNA. Right. So the host DNA is no longer with us, at least isn't intact. Erase the whole thing then here to make it easier. No longer intact bacterial DNA. What we have are little bits and pieces of the bacterial DNA that's left. Okay, so fragments of the bacterial genome. Now, as the viral life cycle progresses, what will occur within the cell is that the proteins that are being expressed by the viral genome are going to start to build new copies of the virus. In addition to that, it's going to build new copies of the viral genome. So there's going to be all these copies of the viral genome. Let's just pretend that's these here. And then there's this a mechanism that pushes the genome into the capsid. So the capsid is the protein shell of the virus, and it'll get transported in there. Now, what should happen is really just the viral genome goes into it. Except in some cases, what's going to occur here, and I'm just going to make this big, just a few of these, is that the viral genome is going to be put into all these. But as they're packaged up, if you remember, there were some fragments and bits and pieces of the bacterial genome, and some of these will get pieces of the bacterial genome. Bacterial gene fragments. Now what will ultimately happen is that this cell, this bacterial cell, will die. It will lyse. So we get cell lysis. And these viral particles will then leave the cell And in some of these viral particles, right, and they'll have the, if they're a bacteriophage, you know, they'll have all the complex viral structures that they need to have, the tail pins and everything else. But they're going to have their own genome, and then they're going to have DNA from a bacteria. Now that doesn't, and that's what we call, you'll look this up, you could see it, you can find some other things online on it. This is the generalized transduction. Although the problem with this is, now this is kind of how this happens in general with the virus, but yet what we're not seeing is where's the transmission of the new DNA. A bacteria didn't get new DNA. The bacteria here was infected by a virus and then destroyed, essentially. This is just kind of the first part of it. To really see the transduction, what has to happen is now this viral particle has to infect a new cell. So we have to have a new cell over here. And then what's going to happen is this viral particle will attach to it. It will inject its DNA and the bacterial DNA into this cell, this new cell. And now what can happen is potentially the same thing can happen over again, all right? Or now some bacteria have mechanisms where they can identify the viral infection, they can identify the foreign DNA, and they can destroy it. So they'll destroy the bacterial DNA. But what may happen is that the bacterial genes we can have recombination. Okay, they can be inserted into the genome then of the bacteria, and this new cell now can obtain new bacterial genes. These are sort of random. So that word is, hopefully you can read that. Let me rewrite it so you can actually see. And that is, because that's the key to this one, is it's random bacterial genes, because remember, as the genome over here was cut into a whole bunch of pieces, you have all these fragments. You know, some of the viral particles don't get any fragments. The others get, you know, not the exact same fragment, but they get different fragments, different pieces. Some of it is useless. It won't be coded for. It won't do anything. Some of them be intact genes that will code for information. So when the new cell gets this, it may get something that it can use. It may bring about new characteristics and qualities to that bacteria. Or the cell may just become infected and die, and nothing really happens with it. But that's but overall, that's the process of what we call generalized transduction. And that happens when a virus enters into a lytic cycle. We get general or kind of random pieces of DNA inserted into the viral particles that they can then pass on to other bacteria. That's the idea. And bacteria then get new genes from a viral infection. Now, the lysogenic cycle is going to be a little bit different. In the lysogenic cycle, what's going to happen is that, I'll draw this here, so this bacterial cell is infected. The viral genome is inserted into the cell. So I'll draw the virus here, it's attached. Okay, the viral genome is inserted into the cell. And now what's going to occur is that this DNA isn't going to be replicated and the genes are not going to immediately be expressed. What's going to happen is the viral genome is going to become inserted into the host genome. All right, so we have the viral genome, and then you have the bacterial genome. Okay, so the bacterial genome and then you're going to have the viral genes here. Viral genes are inserted. So they're inserted into the bacterial genome. And the thing is, they may hide out there, and they may hide out there for a very long time. And what is going to happen then is this cell isn't going to be killed. It's not going to lyse like this one down here. So give me a second here. I'll do a little bit of erasing to give us some more space. So generalized transduction, the cell is infected. The cell pretty much dies quickly, and some of the pieces and particles may or may not get little fragments randomly of the DNA from the bacteria. In the specialized transduction, which is what we're talking about now, that starts off with the viral life cycle that we're going to call lysogenic. Go over here. So now in this lysogenic life cycle, the virus infects the cell, it injects its DNA into the cell, but the DNA is integrated into the viral genome. So say goodbye to lytic and generalized transduction. This cell then may just go on about its business and divide into two new cells. When it does, DNA replication will occur. And when that replication occurs, it'll replicate the entire chromosome, which includes the viral genes. So you're going to get viral DNA replication occurring just as the bacterial DNA is being replicated. And so we just get more copies of the cell, more copies of the viral genome, more copies of the cell, more copies of the viral genome, and this will just continue on and on and on. Okay. So at this point, this particular bacteria, through the viral infection, has picked up new viral genes, which are probably not being expressed and so are not really affecting the cell at all. So again, where does the transduction come in? How does it pick up bacterial genes? Well, what's going to occur now is that this viral genome at some point will be triggered to be expressed. And when it's triggered to be expressed, then it's going to essentially enter into what was the lytic cycle that I had over here that I erased, and now we're going to kind of come back to that. So what's going to occur is this, and this is kind of the important part here, is that the genome is then going to be cut out and removed from the host. But the thing is, when it's removed, it may take with it, and it often will take with it, a little piece of the bacterial gene. So this will then get cut up. So the host DNA, bacterial DNA, is now destroyed. And you just get the fragments of what are left. Except that when that happened, this piece here, which is cut out, excised, will likely cut out with it some parts of the bacterial gene. So we have some bacterial genes joined to the viral genome. Now what's going to happen is those pieces are going to be copied, over and over and over again. And so we're going to get, lots and lots of copies of these here, which ultimately are going to be packaged into viral particles. And they're all going to be exactly the same. Okay, so the same genes go into all viral particles. Different than the generalized transduction. There we're going to get some potential bacterial genes put into the viral particles, but they may not get any, and if they do get any, they're probably going to be all different pieces of DNA. Here, this is the exact same set of genes in all these viral particles. Now what's going to occur is this. These viral particles will leave and then infect a new cell. So let's say that's this cell up here. We'll come back to this. Let's say now this is our new viral particle. This is a new cell, let's just say. Maybe I won't do that. For the sake of this whole drawing up here, I'm actually going to draw a whole new cell. I'm just short on space, so I'm just going to have to take a second here to do this a little bit more efficiently. So we're going to get a new cell here. Here's a new cell. This is a bacterial cell. This bacterial cell has its own DNA, has its own chromosome. But now it's going to be infected by one of these viral particles. And what's going to happen is it's going to inject into the cell its genome. So it's going to inject the viral genes and the genes from this other bacteria. And then it's going to essentially go into the same thing. What's going to happen is, essentially, I could do this over here, this segment is going to get inserted into the genome. Now the thing is, it's different this time. Okay, this time this cell here, is going to then replicate and so on, and it's going to replicate then with it now genes from a different bacteria. But in addition to that, it may express some of those bacterial genes. Okay, so when that happens, and this gets genes from a new bacteria, it receives genes from the virus and another bacteria. It may be able to express those bacterial genes. So this is going to be a little bit different here now, right? What you have is a virus carrying with it viral genes and bacterial genes from a previously infected host cell. And then when that virus then infects a new cell, it's going to give it its own viral genes, but it's also going to give it bacterial genes from the previous organism. And because of the process, this lysogenic process, where the DNA is inserted into the host's chromosome, that chromosome could then be read, and those genes from the other bacteria can be read, and they can be expressed. So then we add to the genetic diversity of this infected bacteria as it's just going about its business, doing DNA replication, doing gene expression. So this is replication plus gene expression. And now, after being infected by one of these types of viral particles, it can be expressing genes from different bacteria. So we've actually altered the diversity. And again, if this bacteria has a mechanism that can identify foreign viral DNA and clip it out, it could potentially then cut out just the viral genes, maybe, sort of its own bacterial immune system, and get rid of them and destroy them. But it will most likely leave the bacterial genes behind, the ones that came from a different organism, and now it's not really infected, and it is gained new genes from a new organism. So there's a lot of maybe, maybe not. This may happen, this may not happen. The information may code for something, it may not code for something. But over time, with all the different types of viral particles, with all the different combinations and all the pieces, bacteria do successfully get new DNA integrated into their genomes and then continue to hold on to that DNA and then change over time. All right? And this is one mechanism that allows that to happen through viral transmission, right, of an infection. So that's the last of these types of horizontal gene transfer. So it's a transfer of genes, in a way, this is transfer of the genes between, this bacteria and this bacteria over here. This bacterial cell, when it was infected by the virus, that virus essentially took with it little pieces of its DNA. And then when it infected a new cell, it gave that cell pieces of the DNA from a different bacteria. Right? So that's our transduction. We had conjugation, where two bacteria, one has the ability to pass DNA to another bacteria through the plasmid. And then we have transformation, again, where it's just coming from the environment, just random sorts of pieces. So those are the main mechanisms, in addition to mutation, that contribute to diversity within the genomes of bacteria.